Hi, my name is Grace Connelly. I'm in the Ellison Lab studying cover cropping in hemp. I'm specifically interested in how we can limit the amount of added nutrients in hemp production. So using clover as a way to increase the amount of nitrogen present in the soil that the plants can take up instead of having to add nitrogen synthetic fertilizers. I'm looking at plant health, and especially I'm looking at soil health and the soil microbiome and kind of trying to see a broad picture of how cover cropping could be a possible alternative cropping system in cannabis production.
Hi, my name is Sophie Perry, and I work in the Godot Lab in the Department of Entomology at UW-Madison, and I am studying hemp integrated pest management. We have a trial here at Arlington where we are looking at 4 different cannabinoid hemp varieties, and we're comparing borer pest damage across those 4 varieties and also looking at how borer pest damage changes the cannabinoid content of these cannabinoid hemp varieties. So we are counting larvae every week of 3 main borer pest species, which is the European corn borer, Eurasian hemp borer, and the corn earworm. We're trying to see how their different infestations are affecting yield, as well as how at the end of the season we'll take some flower samples and analyze them for CBD and THC content to see how different levels of infestation might affect the CBD and THC content of these different varieties. We also want to see if there's a variety that has more resistance than others to these different borer pests. So if we find one with significantly less of a borer population, that could be something to implement for pest prevention for growers.
Hi, I'm Mike Petersen. I work at the Wisconsin Crop Innovation Center out in Middleton, Wisconsin. Our main focus with cannabis is we're genetically engineering cannabis and editing cannabis to introduce new traits into the crop. Some of the things we've done are we've created a THC-free line, so totally free, never will go over 0.3% THC. We're working on a powdery mildew-resistant line, which we've got great results for so far. And then in addition to those 2, we've got all kinds of putative lines that may have minor cannabinoids in them, different terpene profiles, different flavonoid profiles, other disease resistance, maybe yield, fiber qualities, lots and lots of different traits that we're working on and continue to look into. So.
Now are you?
So we're in the grain and fiber trial here. Kind of similar to what's going on over there. We're going to talk about the variety trial, but I'm going to talk about the breeding program a little bit. So when we started this program, my program started in 2020. There was no hemp seed available. I did not take over a hemp breeding program. So the first thing that we had to do is get seed to start researching hemp. We didn't have any. So one of the big efforts that our lab has taken on is finding seed that we can use in our research program and in our breeding program. And part of that is what's available to us in the Midwest, and that is feral hemp or ditchweed that was left over from when Wisconsin was one of the largest hemp producing regions in the country back in the 30s, 40s, and 50s. And so we went around to collect that material, and we're going to hear a little bit more about this, but we've collected feral hemp across 12 different states, and that's the basis for the breeding program and the research program. And that material has been surviving here, adapting to this region, and is an excellent source of really interesting genetic diversity for the pests that are in the region, the different stress, climatic stress, the climate we have here. So that's part of what we're doing now, is taking that material, selecting for that, finding the most vigorous plants, breeding out the weedy traits. We don't want things like seed shattering at the end of the season. So we select against those type of traits. We don't want small seed, we want larger seed. We just select the best and then combine them we work to understand the genetics of those traits. And yeah, it's a lifelong process. So I'll be doing this hopefully in another 30 years. You'll see some really cool Wisconsin varieties of hemp, but it's really fun because it comes from this material that we were able to source and collect in the region. And we're going to talk about how that germplasm that has been the basis of this program is being used in a bunch of different projects. So I'll let people speak about those individual projects, but it's neat because it's really something that feels local. It's been here. We were able to collect that and now utilize that again. So it feels very cyclical.
So I collected feral hemp plants across for two summers of 2022 and 2023, and I still have materials coming in. And all in total, I was able to collect from 1,800 plants, seeds and leaf tissues from all of these plants. And we grew all of these plants in the field last year, and then we characterized them for some unique and specific traits, which include like flowering time. Then we were able to see some specific unique abilities in terms of the seed size and all of this. And so just to draw back a little bit, so we characterize those materials phenotypically on the field, and then we also genotype these materials using paste assay markers, and we did this to be able to distinguish the feral hemp population into the three profiles that we know, that is the THC types, the intermediate types, and then the CBD types. And then we also went ahead to chemotype them using GCMS, gas spectrophotometry, to quantify the amount of CBD and THC each of these plant materials had, and also 9 to 11 other cannabinoids in all of these plant materials. And these materials were also a subset of all of these collections, like 760 thereabout, were sequenced at UW Biotech Center, and this was to understand the level and amount of genetic diversity that was present in these feral hemp collections across 12 states of the United States. The big objective of this project was to help build and populate the newly established feral hemp germplasm repository at Connelly Agritech with diverse and compliant plant materials that could be useful for plant breeders across the United States, knowing fully well that this was really a big problem to access feral hemp germplasm for research after the 2014-2018 Farm Bill. And so today we have some of our materials that are undergoing evaluation for variety release trials, and this is a project in collaboration with University of California, Davis, the Ag Center in Cornell, and also UW-Madison and some other institutes. So they're evaluating these varieties for sex, flowering time, and also for some diseases like sectorial and powdery mildew, but I think that's going on in California. And so it's really exciting to see that some of our materials are already being used, and some of these varieties should be, some of these materials should be released for breeders maybe early next year.
This has been funded for the last 3 years via the USDA Specialty and Alternative Crops Grant Block Program. And this is a collaboration between 4 universities across the Midwest: UW-Madison, Michigan State, Purdue, and the University of Illinois. And so what we do is we replicate these variety trials across the Midwest to find out which varieties grow well, which grow poorly, and for which purposes. I think something that's very fascinating that Ademola talked about, and this kind of anecdotally, was that 80% of the samples were compliant for THC from those feral collections, and I would be shocked if that number was very different in these fields as well. Industrial hemp for grain and fiber typically produces much lower THC or CBD. The CBD trials that we saw, the check program that we just came from, about 80% of those crops have potential to fail if not harvested at the right time. So we are talking about 2 very different plants, okay? So when we talk about the challenges that growers are facing, every grower has to grow compliant hemp, but the inherent nature of these plants to produce those is typically much lower. So the burden on the grower is much higher. So anyway, we have 3 different types of trials out here. We have our fiber type, which is resulting in some 12, 13, 14 feet tall plants. We have our dual purpose and our grain, as Shelby mentioned. I would just like to point out to you that if there is a sign in front of the plot, it is a representative of the variety that's in there. There is a bunch of other plots of that variety, which you can find in here, but we wanted some that were right out in the front for you. And if you walk actually into the second and third rows behind, there's going to be some more signs for other varieties that did not get planted in their block in the front. So some of the things I'd like you all to know is that, depending on how you plant these varieties and what their intended use is going to be, your production system, your practices are all going to be very different. For our fiber varieties, some of the varieties were ready and to be harvested, so we already did, and that's why there's no plants there. But what we're trying to do right now is cut these materials down, evaluate them for fiber quality, and then on the grain side, what we're trying to do is find varieties that grow well here, that do not shatter, that we can get high yields on, and that ultimately don't mold or spoil and can be having high potential for use in a variety of applications. But the basis of this trial is looking at yields, and we spent 3 years trying to find the best varieties that grow well here. The future is about finding the best ones and the ways that we can grow them to produce real products. And so this is a baseline for the next step. It's what grows well here. How do we breed varieties that can go into a commercial breeding program and compete with these varieties, and then actually using them for materials.
So I'm Derrick. I'm a postdoctoral researcher in Shelby Ellison's group. I work on plant pathogens of cannabis. So in particular, for this field day, I've been talking about Fusarium head blight on sort of grain varieties, which you can kind of see right here. And then we are working on researching different methods of mitigation. So whether or not that's cultural practices, such as like planting resistant varieties or researching resistant varieties, or controlling inoculum through different seed treatments. And so that's sort of what it occurred.
It's not many years ago now that we didn't have really any pest management tools in hemp. We're starting to get a little trickle working down. I just want to give you a very brief update on some things that are being worked on at IR4 and things to expect coming down the pipe. So hopefully some of this means something to you if you're a little familiar with these products. The first one is the active ingredient abamectin. This is sold as Abba Ultra and other products for mite control. The field research is completely done for that one. All of the samples from around the United States are sitting at the lab. And you're going to hear that as a very common thing for several of these projects, because the lab now needs to develop specific protocols that EPA is going to accept the data from, right? So they're going to look at both the CBD oil that's been collected, or the buds that have been sent in, as well as the grain and fiber that's been sent in, and analyze them for residues of the pesticide to see whether it can meet the EPA tolerance for that. The second one is chlorantraniliprole as the active ingredient. That's sold under the trade name Coragen. That would be primarily for lepidopterans, aphids, and so forth. That one is also, samples are done and at the lab. Cycloniliprole, sold as Harvanta, again for leps, aphids, thrips, whiteflies in the greenhouse. That one is sitting at the lab as well. Fenpyroxamate, which is sold as Portal for mite control. That one is also done in the field and sitting at the lab. Flutionil, which is sold as Gatton, primarily for powdery mildew control. That one is actually very close to submission to EPA. The lab has completed analysis of those samples. The final report is being developed, and that'll be submitted to EPA. Once we're at that point, we're maybe within 1 to 2 years of possibly seeing a label on it. Okay, so that one's getting a little closer. Isofetamid, or Kenja, for numerous pathogens, is also at the lab. Quizalofop, or sold as Assure 2 for grass control. That one is also getting very close to submission. I wouldn't be surprised if we're 1 to 2 years out also from seeing some sort of label on that. Zetacypermethrin, or Mustang Max, again, a insecticide primarily for leps, thrips, weevils, is also done in the field and at the lab. So we have a lot of things sitting at the lab. The final one I'm going to mention is the active ingredient flonicamid, which is sold as Belief. That is a current 2025 project. So we have trials here in Wisconsin. There are 2 locations here on the Arlington station. There is one at the Hancock station and one at the West Madison Ag Research Station, where we are getting close to actually sampling, collecting those harvests and getting those sent off to the lab. So there's a lot of things in the pipeline. It's not a quick process. I've told you that all along, but we are starting to see that progress coming out the back end. And I think this backlog that we're seeing at the lab, we're going to start to clear pretty quickly.
Hi, I'm Jill McGhee. I'm a third-year PhD in the Ellison Lab. I work predominantly on the genetics, the genetic differences between feral and cultivated hemp, mostly in the germination seedling establishment sector. So really trying to get seeds that germinate all at the same time so you get uniform flowering and you get uniform harvest times.